Okay, so there was a bit of a network issue due to which the link was showing some problems. I think that's why some people might be late. I'll start the session and we'll see to it whenever someone joins, I'll let them know. I'll share my screen. So, hi, is my screen visible? Can you please comment, is my screen visible? Okay, one second, I'll check. Yes, yeah, so I'll start the session. So good evening, good afternoon. This is Aishi, and last Friday I was actually supposed to take a session last Friday, but due to some problems, I had to postpone the session. So I am a PMRF TA for the course Applied Environmental Microbiology, and today we'll be discussing some of the sample questions from the course that will help you solve other questions from the assignment of this year. So these are some of the past assignment questions and some of the last year assignment questions as well. And I'm currently pursuing PhD in the Department of Chemistry, IIT Bombay. So we can start the session, and I'll let people in as and when they come in. So moving on to the first question, with respect to ruminants, which of the following are true? They are mostly hindgut fermenters, as it's a highly efficient method. In the experiment with 15 nitrogen labeled urea, most of the labeled urea was excreted out as compared to foregut fermenters. Both are true and both are false. So you can take a moment to think, otherwise I'll explain what the answer might be. Okay, so the answer is both are false. But before that, we'll learn a little bit about hindgut fermenters and what does the term mean. So a challenge that is common to all the herbivores is the processing of tough plant tissues, notably high in lignin or cellulose, because cellulose is a polysaccharide and it's the major structural component of the plant cell wall, and it cannot be hydrolyzed by the endogenous enzymes of vertebrates. It's only after symbiosis with cellulase producing microorganisms that sufficient nutritional content can be gained from this food source. So herbivorous mammals all utilize fermentation chambers in some portion of the gastrointestinal tract to maximize exposure of fibrous foods to these digestive bacterial agents. The fermentation chamber may be situated in the foregut area of the stomach, that is, for for example, ruminating hippos or like sloths or rodents or kangaroos, or in the hindgut area of the stomach. Um, hindgut area of the cecum colon or colon area, basically, such as monkeys or caviomorph rodents, rabbits, wombats, etc. And there was a study that was conducted that was basically concerned primarily with the differences between ruminating artiodactyls, which are the only foregut fermenters, to regurgitate their food, like how they chew their food, and there's another perissodactyls that were horses, rhinos, and tapirs. So there's an opportunity for a confusion of, in this case, for the terminology, because the term ruminant can either refer to physiology, that of the foregut fermentation combined with gut chewing, or to a phylogenetic grouping of artiodactyls that excludes camelids. So in this, for this question, we'll use the term ruminant in the physiological sense here to include camelids. So as you can see from here, hindgut fermenters have a shorter passage time than ruminants, and hence are very less efficient in cellulose digestion. Cellulose digestion, and hindgut fermentation basically takes place in the colon and additionally in the enlarged cecum, which act like fermentation chambers in much the same way as the rumen does in ruminants. So this arrangement presents the problem that the cellulose is not fermented until this point, and the volatile fatty acids must be absorbed in the colon rather than in the small intestine. So hindgut fermenters have a shorter passage time than ruminants, and hence are less efficient in cellulose digestion, for which they compensate with a higher intake of food. Note that an additional problem for hindgut fermenters is that they must access the cell contents of the herbage prior to the fermentation of the cellulose in the hindgut. And although the products of the cellulose fermentation can be absorbed in the colon, the enzyme producing glands for the digestion of the sugars, fats, and proteins of the cell contents are located in the small intestine. And some non-ungulate hindgut fermenters, such as rabbits and certain rodents, they circumvent this problem by refection, that is, eating the initially produced feces. However, refection is not practiced by any ungulate, nor by hyraxes or elephants. Thus, it must be the case for hindgut fermenting ungulates that the initial mastication of the food is sufficient to fracture the plant cell walls to release the cell contents prior to the site of cellulose fermentation. Thus, hindgut fermenters basically face two functional challenges with food combination, in which they differ from ruminants. Not only must they consume more food per day than a ruminant of similar size and diet, but they must also ensure that the cell walls are ruptured on initial food ingestion. One would therefore predict that initial food mastication would be more prolonged and intensive in hindgut fermenters than in ruminants. And even the ruminants later regurgitate their food and chew it as gut. At this point, the food has been softened by fermentation processes and may present a reduced load on the masticatory system. So morphological studies do appear to show that hindgut fermenters have deeper jaws, larger areas for the insertion of masticatory muscles, and greater cheek tooth occlusal area than ruminants. And this would accord with the hypothesis that hindgut fermenters experience a greater load on the masticatory system than ruminants. And with regards to food ingestion and digestive physiology in general, the usual comparison between hindgut fermenters and ruminants is between horses and cattle, both medium to large sized ungulates with a similar diet of grass, and both animals have been the subject of many agricultural studies as such. And while other ruminants have also been studied in the same fashion, like sheep, deer, or llamas, studies of other extant hindgut fermenting ungulates such as rhinos, elephants, hyraxes are few in number, and detailed comparisons of food intake behavior between horses and cows are rare, although horses do seem to show longer grazing times or higher food intake than cattle. In addition, also it has been shown that while ruminants initially swallow large particles which are later reduced in size by rumination, the fecal particle size in horses is relatively small, despite the fact that they only chew their food once. And there was one pilot study that has directly compared the ingestion behavior of horses and cows. So this study suggests, although it cannot statistically be demonstrated, horses chew their food longer than cows on initial ingestion, and thus this difference is more pronounced with forage of increasing fiber content. So as these are some of the differences between foregut and hindgut fermentation process. For foregut fermentation, the cell solubles are inefficiently used. However, for hindgut fermentation, these are more efficiently used, but the microbial protein is lost, which is recaptured in case of foregut fermentation. Then there's thorough digestion of cellulose. There less thorough digestion of cellulose, and foregut fermentation is either moderately abundant with fibrous forage, or hindgut fermentation is less abundant with higher quality food, or more abundant with less fibrous food. So this, based on the anatomy, biochemistry, and products of digestion, these are some of the other differences between ruminants and hindgut herbivores. Biochemistry wise, the digestive enzymes are of microbial origin. It is the same in case of ruminants. And the products of the digestion which are absorbed are volatile fatty acids from the rumen. However, in hindgut for herbivores, it's the limited glucose and amino acids from food. But the microbial protein is lost, which is recaptured in case of ruminants. So next question, first generation sequencing is more accurate than second generation sequencing technique. Is it true or false? Anyone? Take a moment to think about it then. So just try to remember what the first generation sequencing entails, what the second generation sequencing means. Okay, so I'll explain. The first generation, it's true. The first generation sequencing is more accurate, which is the Sanger sequencing, for which Sanger actually got a Nobel Prize. This is more accurate than the second generation sequencing technique. So these are some. This is a schematic to understand the differences between the three techniques. So the first generation Sanger sequencing basically relies on the selective incorporation of chain terminating dideoxynucleotides by DNA polymerase during DNA replication. So we know how DNA replication occurs. So there is a OH group which is further taken care of by the next incoming nucleotide. But if there is a dideoxy nucleotide that is coming in the place of your normal deoxynucleotide, that doesn't have any OH group, it has a H group at that point, and the next nucleotide cannot come and attach to it. So that is why the chain gets terminated. So during the early days of this technology, dideoxynucleotides were radio labeled to allow for detection and manual determination of the consensus sequence. However, applied biosystems later introduced fluorescent base labeling, which allowed automation of the process and subsequently higher throughput. And automated Sanger sequencing is a sequencing platform of choice for almost 20 years and was used to sequence the first finished human genome. And in 1995, the first bacterial genome sequence for Haemophilus influenzae was published, and it was sequenced by the Sanger method. Specifically in this approach, genomic DNA from this Haemophilus influenzae was isolated, randomly sheared, that is broken down, and cloned into a plasmid vector. And the resulting vectors were transformed into host E. coli cells and propagated. Plasmid DNA was extracted, and insert DNA from Haemophilus influenzae was sequenced from both ends. It was called random shotgun sequencing, and this produced a sequence lengths of about 460 to 480 base pairs per clone. And this was a monumental task and required the production and sequencing of almost 9,000, 10,000 E. coli clones to obtain a draft genome with 5x consensus coverage. And this work also knowingly left 37 of the genome unsequenced. Despite this method being very time consuming and labor intensive, hundreds more bacterial genomes, including those of the Mycobacterium tuberculosis, Yersinia pestis, E. coli K12, Bacillus subtilis, Treponema pallidum, all were sequenced this way over the next few years. And the relatively high throughput of the automated Sanger sequencing led to the birth of bioinformatics as a mechanism to assemble the shotgun sequences into genomes, and later to help make sense of rapidly accumulating sequence data. So during the first generation of microbial whole genome sequencing, there was a strong emphasis on closing or finishing genomes, and the first software packages were designed to assist in this endeavor. And the first generation assembly programs such as Alora, Celera, Assembler were used as an overlap layout consensus algorithm and were optimized to work with the high accuracy long reads produced during Sanger sequencing. And the first bacterial genome, that was the Haemophilus influenzae, was closed after initial assembly with the TIGR assembler into 140 contigs with 98 sequence gaps, and various other algorithms were further then used to map and fill the physical gaps, while sequence gaps were closed by primer working. So this has a very high accuracy. The read lengths are really long, and that's why it's still one of the sequencing techniques of choice. So the basic technique is what I've described in one of the previous classes, is that a dideoxynucleotide comes and takes the place of a deoxynucleotide, so the chain gets terminated and no further chain nucleotide incorporation occurs. So there are 4 tubes, and 4 tubes have different dideoxynucleotides. If one has dideoxy adenine, some has the next tube has dideoxyguanine, and so on. So in that, and the same sample is placed over there. So all the 4 deoxynucleotides are present in all the 4 tubes, but each tube has one specific dideoxynucleotide, and when that dideoxynucleotide gets into the place, the chain is terminated. In the second tube, since another kind of dideoxynucleotide is present, it will get terminated, it will get incorporated when the sequence that we are trying to read has a complementary base. So in that way, we can actually, the different chain lengths, different chain termination will occur at different chain lengths in all the 4 tubes, and then we can run a gel and see which chain lengths were terminated at which point, and by that we can figure out which tube it came from, and that particular nucleotide went into that sequence, and by that we can figure out the sequence of our generated strand, and from that we can obviously figure out the complementary strand, which was the strand we were trying to read. Then we have the second generation, which is a massively parallel sequencing technique. So this was first introduced by Roche in the form of 454Gs20 in the year 2005, and was followed by Illumina's genetic analyzer in 2007. So the early versions of the next generation sequencing platforms came with several hurdles to routine whole genome sequences of organisms. For example, for a short sequencing reads 110 BP and 35 BP require the development of novel assemblers optimized for the assembly of short read sequences. And in addition, each of these platforms had a massive footprint and price tag. However, by 2010, benchtop versions of these sequences came into the market and were available by most microbiology labs, leading to an explosion in the number of microbial sequences available for comparative analysis, epidemiological investigations, ecological studies, and so on. So each next generation sequencing platform had a slightly different approach to DNA preparation, sequencing, imaging, and analysis, and variation in the approaches resulted in each platform having different strengths and weaknesses. The 454 platforms produced longer reads, which improved mapping in the repetitive regions. However, they suffered because of high reagent cost, high error rates in the homopolymer regions, and produced less data per run than their competitors. So the major shortcoming of all the second generation sequencing platforms is relatively short read length, and genome assembly algorithms built for the assembly of Sanger data do not perform well with the short read data produced by second generation sequences. And in addition, the closed genomes produced during the first wave of microbial sequencing can now be used as a scaffold on which to guide the assembly of short read data, for the first time giving scientists the choice between de novo and reference guided genome assembly methods. So de novo genome assembly can be done by a variety of assemblers. Reference guided assembly maps short sequence reads by assessing the placement of each read against the reference genome and calculating the probability of its match with the reference. So these alignments are then used to construct a novel consensus sequence for the sequence data. So the second generation sequencing gave rise to the proliferation of the available draft genomes, where genomes that are sequenced to a high coverage depth but not close are submitted to databases and are used for downstream analysis. And this has generated an ongoing debate in the field of microbial genetics about the allocation of resources. Is it better to close a lower number of genomes, or should a higher number of genomes be sequenced to a draft status? Because draft genomes are sufficient for the majority of analysis, including virulence factor identification, phylogenetics, and MLST. However, closed genomes are required for genomic island identification, characterization of repetitive elements, and sometimes structural rearrangements. And then comes the third generation sequencing technique, the single molecule sequencing, which can address the problems inherent to short sequence reads by sequencing long single molecules in real time. And this was probably introduced by Helicos Biosciences Corporation and was initially used to sequence a viral genome, but quickly progressed into parallel sequencing and the sequencing of the human genome. However, the short reads produced by the Helicos platform did not differentiate this technology from its second generation counterparts, and it was no match for the single molecule real-time sequencing platform that later PacBio introduced. Pacific Biosciences basically, it was able to regularly produce reads exceeding 1 kb. So, however, what the second generation sequencing lacked in sequence read length, third generation sequencing appears to lack in accuracy. And in comparison, the second generation Illumina MI sequence platform has an error rate of 0.58%, and the Pacific Biosciences platform's error rate is 14%. So, despite the rapid emergence of all these technologies, first generation technology still remains one of the most highly accurate systems to actually figure out the sequence of the DNA. So that's why first generation sequencing is still much more accurate than second generation sequencing technique. So it's true. Moving on to the next question. Wild type microbes are different from the laboratory bacteria in sense that they do not undergo any mutation. What do you think? It's true or false? Take a moment to think about it. So when we use the term wild type, we by default refer to the system that generally is standardized in the environment, or that is most common in the environment, that doesn't have any changes in its genome or anything, and then we go and refer others as the mutant strains which have differences in the genomes with respect to these wild type microbes. So with that respect, wild type microbes do not have mutations, but then they have used the term laboratory bacteria as well. So laboratory bacteria, if we decide to incorporate any mutation in a wild type microbe, then that obviously becomes a mutant strain. So with respect to that, this statement is true, because wild type microbes are different from the laboratory bacteria, because if the laboratory bacteria is a mutated version that has been made in the lab, then obviously it's a mutated one, but the wild type microbe does not have any kind of mutation. Otherwise, this might be a false statement as well if a different kind of explanation is derived, and I'll explain that. So our present in-depth knowledge of the physiology and the regulatory mechanisms of microorganisms has risen from our ability to remove them from their natural complex ecosystems into pure liquid cultures, right? So these cultures are grown under optimized laboratory conditions, and these allow us to study microorganisms and individuals. However, microorganisms naturally grow in conditions that are far from optimal, which causes them to become organized into multicellular communities that are better protected against the harmful environment, like biofilms. So, and moreover, this multicellular existence allows individual cells to differentiate and acquire specific properties such as forming resistant spores, which benefit the whole population. So, the relocation of natural microorganisms to the laboratory can result in the adaptation to these favorable conditions, which is accompanied by complex changes that include the repression of some protective mechanisms that are essential in nature. So, laboratory microorganisms have been cultured for long periods under optimized conditions and might therefore differ markedly from those that exist in the natural ecosystems. So, laboratory strains are like cultured in optimal growth media, which has optimal nutrient concentration and everything required. So, even if they are the wild type without any mutation, they still will vary a lot from the wild type microbes that are actually found in nature, because in nature it's very far from optimal. Even though they do not have any mutations in the genome, might be both of them, but still they'll differ markedly, because differ significantly, because in case of laboratory strains, they are grown in a very perfect environment. There are nutrient replenishings happening, that we are changing the media every time it becomes confluence and everything. So that's how, even though keeping the mutation perspective out of this conversation, even then these two strains might not be the same. And for this I have picked out an example. So this is the same bacteria, but this is a laboratory strain of Saccharomyces cerevisiae, and these are the wild strains of Saccharomyces cerevisiae. So you can see the huge difference in the morphology, in the appearance and everything, and I'm pretty sure there'll be some differences in their inner mechanisms as well. So even though they are the same strains, and probably they do not have that kind of genomic mutations happening, genetic mutations happening there, but still these are very different, because one has been grown in the laboratory under very specific optimal conditions, the other has not been. And as you know, that microorganisms are very traditionally very good models for the investigation of the basic cellular processes on a unicellular level. However, these seemingly unicellular microorganisms naturally form multicellular communities, and they differentiate into specialized cells that benefit the whole population and synchronize their behavior under certain conditions, basically biofilms. And wild microorganisms are able to modify their properties according to their nutrient supply, and in particular are able to adapt to the limited nutrients. It's basically they have very limited nutrients, but they adapt themselves according to that environment. This is a very common situation in nature, and microorganisms are able to decrease their rate of metabolism and survive using rare nutrient sources, including their own waste products. For example, ammonia signaling in yeast colonies. Some of the mechanisms that are apparently very important in nature, such as the formation of extracellular matrix, are switched off when microorganisms are transferred to comfortable laboratory conditions, whereas others that appear to be more general persist. So some of the general mechanisms, they actually stay inbuilt in the system, but to survive in the harsh environment, as we said, they form multicellular communities called biofilm. They come together and form a slimy structure, but this mechanism is apparently in some cases gets switched off when they are grown in the laboratory environment, because in laboratory environment there's unlimited supply of nutrients because people are replenishing the media every time. So there are proper papers on the internet, if you want to look, if you find this fascinating, which discusses the properties of multicellular microorganisms, including their differentiation, interaction, long range signaling, and considers how these properties differ in the microorganisms that are routinely studied in the lab. And also multicellular microorganisms have advantage in nature. In the lab, under conditions of optimal nutrient sources, temperature, humidity, and so on, microorganisms are usually treated as individuals and are studied predominantly during the relatively small window of time in which they grow as exponential pure liquid cultures. However, the natural environment rarely provides microorganisms with conditions that allow them to grow and reproduce at a maximal rate. Microorganisms in the wild need to cope with, for example, nutrient starvation, temperature changes, and a lack of moisture, and should be able to survive for extended periods without reproduction. In addition, microorganisms rarely exist naturally as individuals because one of their survival mechanisms is the ability to organize themselves into multicellular communities and differentiate into specialized cell variants. So traditionally studied fruiting bodies of slime molds and myxobacteria are formed by motile amoebas and bacteria under starvation conditions, and the cell differentiation that occurs during the development of these structures results in the formation of highly resistant spores and fruiting body stalks that finally lies. So the environment of the fruiting body seems to drive the development of mature spores that are able to survive in the extreme environments. And during this period, the material released from the live stalk cells is incorporated into the spore cell wall, which makes the spores fully resistant to harsh conditions. So all these findings are there in published papers, and these reveal the existence of differentiation programs that are specific to distinct cells and areas of the fruiting body and which cannot be pursued by individual free living cells. So that's why there are several indications that environmental conditions have an important influence on the properties of the multicellular communities that are formed by microorganisms, and this is particularly evident in biofilms and colonies of bacteria and yeast. So complex social communities of different coexisting microorganisms are common in nature, and these highly organized biofilms can attach to solid surfaces in liquid environments or form a pellicle on the surface of water. And indeed, their formation is so efficient that it is an arduous task to keep surfaces free from an invading biofilm. But contradictory to that, in the lab, it is difficult to create artificial experimental conditions that stimulate natural conditions. In other words, to grow artificial biofilms that have properties similar to those of fully developed natural biofilms. It's very challenging, and therefore, despite the fact that there is plenty of information about the occurrence and ultrastructure of biofilms in situ, relatively little is known about the ecology and biology of cells within biofilms and about biofilm dedicated pathways, because it's almost very challenging to reproduce these things in the lab. So, and colonies are another example of multicellular communities of microorganisms that occur under natural conditions. They seem to be less complex and less resistant to the environment than biofilms, and therefore represent an intermediate between unprotected individual cells and fully protected natural biofilms. So this, together with the fact that colonies of bacteria, yeast, and other microorganisms are commonly grown in labs, this makes them an interesting subject for the studies of the interactions and signaling between microorganisms within multicellular communities. So one very fascinating question is whether these colonies in nature differ from those growing under lab conditions. So recent findings on the differences between colonies found by these Saccharomyces cerevisiae lab strains and those formed by strains freshly isolated from nature. These are the lab wired strain. This is from the lab. These have provided some clues. So Saccharomyces cerevisiae lab strains usually form smooth colonies with no structured pattern, whereas colonies of wild Saccharomyces cerevisiae strains form fluffy structured colonies even in the lab. So ultrastructural native electron microscopy and biochemical analysis reveal that cells within fluffy colonies are connected by an abundant, highly glycosylated extracellular matrix that forms a scaffold for the colony. So no such material was observed within the smooth colony, the lab strain. So this extracellular matrix is important for the protection of the colony from perilous environmental factors and desiccation. And moreover, similarly to biofilms, it can form small channels for nutrient and water flow, and also chambers and microenvironments for the next generations of cells. So this is supported by the observation that fluffy colonies expand and occupy new territory more rapidly than those that are smooth. So these are some of the differences between lab strains and wild strains. So mutation, even if we keep the perspective of mutation aside, they'll still not be same because of the environmental conditions that they are brought up in. Moving on to the next question. Genotypical differences may or may not lead to phenotypical differences. Anyone? It's a very easy question. Anyone would like to try? Still in the meeting. Let me know if I am not audible or if you are facing any problem with any of the questions. So yeah. So this is true. So genotypical, it's not written, it's not a rule that genotypical differences will always lead to phenotypical differences, because as we know, genotype basically talks about the genetic sequence, and gene basically determines the traits which will be expressed. Organisms that look the same do not necessarily have the same genotype, and biological tests can be done to determine genotypes, whereas phenotype is the physical manifestation of the expressed genes or by the visible traits, for instance hair color, type, eye color, body shape, height, etc. And it depends on the genotype, but it also influenced by some of the environmental factors. So if your body type is there's a genetic component to your body type, you might be having an edge inclined towards obesity if the genetic terms say that. But then also if you are having a very healthy lifestyle, then that might be or your body type might be lean from the genetic things that you have inherited from your parents. But if you have follow a very unhealthy lifestyle with you don't follow anything proper in your diet, or you don't follow a strict exercise regime, then that might lead to obesity in the future. So even though phenotype is determined by genotype, there's also a lot of environmental factors that play a role in that. So this is a table that talks about the difference between genotype and phenotype. So genotype is basically the hereditary information of the organism. It's in the form of genes in the DNA and remains the same throughout the life. Phenotype, however, it's the physical manifestation of those expressed genes or characteristics that are visibly evident. Genotype, the same genotype might produce the same genotype will produce the same phenotype mostly. But in case of phenotype, the same phenotype may or may not belong to the same genotype. Also, genotype is present inside the body as genetic material, so you need to actually conduct proper tests for that. Phenotype is just the external appearance, so you can actually look at something and be able to say a little bit about the phenotype. Genotype is inherited from the parent, from the previous generation. Phenotype is not inherited from the parent. It's kind of inherited from the parent. The statement is not entirely true. So phenotype is something that you inherit from your parents. People inherit their facial features or most of the things from their parents. They might inherit from their parents, or there might be a genetic component to that, but at the same time it's also influenced a lot by the environmental factors, how you live your life, what kind of schedule you follow, etc, etc. Genotype is affected by genes, phenotype is affected by the genotype and some environmental conditions. I've already explained that. Genotype needs to be if you want to determine genotype, you have to go through tests such as polymerase chain reaction. If you want to see the role of a particular gene. For phenotype, it's basically you can observe an organism and tell about the physical manifestation details that you see in that organism. Then we'll move on to an example that will tell exactly why this answer is true. So here you can see capital B is the dominant gene and small b is the recessive gene. So capital B talks about capital B represents the purple color and small b represents the white color. However, capital B is the dominant one and small b is the recessive one. So if we have both capital B's in an organism, that will lead to purple coloration, definitely. If we have one capital B and one small b, that will lead to a purple coloration because capital B is dominant over small b, that is purple color is dominant over the white color. But if you have both small b, it will be white in color. It will be recessive in nature because both of these are recessive, so there is no purple coloration representative over here. So this shows that even though Like even though these two have the same phenotype, both of them are purple in color, they do not have the same genotype. So that is a clear example that all the organisms having the same phenotype might not come from the same genotype. So this is a clear-cut example that will explain the question. Moving on to the next question. Which type of the reactor uses a fill and draw system to treat wastewater and minimize the space requirement by using the same tank for multiple processes? Anybody? Anybody would like to try? So the answer will be SBR, which is the sequential batch reactor or the sequencing batch reactor. So I'll talk a little bit about the sequencing batch reactor. So it's basically a wastewater treatment technology that uses a fill and draw activated sludge technique. And in this system, the wastewater is pumped into a single batch reactor where it is treated to eliminate contaminants before being discharged. And a single batch reactor can be used for equalization, aeration, and clarifying. Two or more batch reactors are employed in a predetermined sequence of operations to improve the system's performance, and both municipal and industrial wastewater have been effectively treated using these SBR systems. They are especially well suited to wastewater treatment applications with low or intermittent flow rates. So the fill and draw batch operations similar to the SBR are not as new as many people believe them to be, because several full-scale fill and draw systems were in use in the early 1920s. And with the advent of new equipment and technology in the late 1950s or early 1960s, interest in the SBRs were reignited. And SBRs, or the sequencing batch reactors, have been able to compete successfully with traditionally activated sludge systems because of the advancements in the aeration devices and the controls. So what is the working of the SBR? So prior to the SBR, most influent wastewater used to travel via filters and grit removal. The wastewater was then pumped into a partially filled reactor with biomass that has been acclimated to the wastewater contents during previous cycles. And when the reactor is full, it functions as a traditional activated sludge system, but without the constant flow of influent or effluent. And after the biological reactions are complete, the biomass settles and the treated supernatant is removed. The aeration and the mixing are turned off, and at any point during the cycle, excess biomass is squandered. And the mass ratio of influent substrate to biomass is essentially constant from cycle to cycle due to frequent waste. As influent flow rates, characteristics and settling tank underflow concentrations change, continuous flow systems modify return activated sludge flow rates to maintain the mass ratio of influent substrate to biomass. Following the sequencing batch reactor, the wastewater batch may flow through an equalization basin where the wastewater flow rate to the additional units processed can be controlled at a set rate. And in some circumstances, the wastewater is filtered and disinfected after it has been filtered to eliminate any remaining solids. And the solid handling system could include a thickening and an aerobic digester as shown in this figure. So there is another return activated sludge system and a primary or the PS sludge system, which are used in traditionally activated sludge systems and are not really required with SBRs. So there's usually one sludge to deal with while using the SBR, and the use of gravity thickeners before digestion is selected on a case-to-case basis based on the sludge characteristics. So when the vessel is filled with wastewater, an SBR acts as an equalization basin, allowing the system to tolerate peak flows or peak loads in the influent and equalize them in the batch reactor. And separate equalization is required in many traditional activated sludge systems to protect the biological system from peak flows that could wash out the biomass or peak loads that could disrupt the treatment process basically. And it's also worth noting that primary clarifiers aren't usually needed before an SBR in municipal wastewater applications. Prior to the biological system, primary clarifiers are employed in most traditionally activated sludge wastewater treatment plants, and if the total suspended solids or biochemical oxygen demand are larger than 400 to 500 mg per liter, the SBR manufacturer may prescribe primary clarifiers. And to assess whether primary clarifiers or equalization are recommended prior to an SBR for municipal and industrial uses, historical data should be analyzed and the SBR manufacturer consulted. and depending on the downstream procedure, equalization may be required after the SPR. And if equalization is not employed before filtering, the filters must be sized to receive the batch of effluent from the SPR, resulting in a large filtration surface area. So equalization is employed between an SPR and downstream filtering because sizing filters to take these batch flows is usually not possible. So this is the entire process of sequencing batch reactor with the diagram. If you're interested, there are actually very nice publications regarding this also. You can check those out. So this is how the solid handling or disposal process occurs. This is the basic mechanism of the equalization as I was talking about, and how it moves the head to filtration and then disinfection, and then the effluent is treated out. So this is what happens in a single batch, a sequencing batch reactor. Sorry. Moving on to the next question. Which of the following is required for the enrichment of chemolithoautotropic nitrifying bacteria? Anybody? If you would like to try. So it's all of the above. What does chemolithotropy mean? So chemolithotropy is the oxidation of inorganic chemicals for the generation of energy, and the process can use oxidative phosphorylation just like aerobic and anaerobic respiration. But now the substance being oxidized, or the electron donor, is an inorganic compound, and the electrons are passed off to carriers within the electron transport chain, generating a proton motive force that is used to generate ATP with the help of ATP synthase. We have discussed all of these. And chemolithotropes use a variety of inorganic compounds as electron donors, with the most common substances being hydrogen gas, sulfur compounds such as sulfide or sulfur, nitrogen compounds such as ammonium and nitrite, and ferrous ion. There are hydrogen oxidizers that the organisms oxidize hydrogen gas with the use of a hydrogenase enzyme, and both aerobic and anaerobic hydrogen oxidizers exist, with the aerobic organisms eventually reducing oxygen to water. Then there are sulfur oxidizers. As a group, these organisms are capable of oxidizing a wide variety of reduced and partially reduced sulfur compounds such as hydrogen sulfide, elemental sulfur, thiosulfate, and sulfate, which is basically frequently a byproduct of the oxidation, and often the oxidation occurs in a stepwise fashion with the help of the sulfide oxidase enzymes. Then we have the nitrogen oxidizers. The oxidation of ammonia is performed as a two-step process by nitrifying microbes, where one group oxidizes ammonia to nitrite and the second group oxidizes the nitrite to nitrate, and the entire process known as nitrification, and is performed by small groups of aerobic bacteria and archaea, often found living together in soil or in water systems. Then we have the iron oxidizers. These organisms oxidize ferrous iron to ferric iron, and since ferrous, or the Fe2 plus, has such a positive standard reduction potential, the bioenergetics are not extremely favorable, even using oxygen as a final electron acceptor. So the situation is made more difficult for these organisms by the fact that ferrous iron spontaneously oxidizes to ferric iron in the presence of oxygen, and the organisms must use it for their own purpose before that happens. Then we have the electron acceptors. So chemolithotropic can occur aerobically or anaerobically, and just as with either type of respiration, the best electron acceptor is oxygen to create the biggest distance between the electron donor and the electron acceptor. Using a non-oxygen acceptor allows chemolithotropes to have a greater diversity and the ability to live in a wider variety of environments, although they sacrifice their energy production definitely. Next, if we come to the amount of ATP generated, just as both the electron donors and acceptors can vary widely for this group of organisms, the amount of ATP generated for their efforts will also vary widely. They will not make as much ATP as an organism will make using aerobic respiration, since the largest delta E is found using glucose as an electron donor and oxygen as an electron acceptor. But how much less than 32 molecules of ATP created depends upon the actual donor and the acceptor being used. So the smaller the distance between the two, the lesser ATP that will be formed. Then we can talk about the difference between chemolithoautotropes versus chemolithoheterotropes. So most chemolithotropes are either autotropes, where they fix atmospheric carbon dioxide to assemble the organic compounds that they need, and these organisms require both ATP and the reducing powers such as NADH or NADPH in order to ultimately convert the oxidized molecule CO2 into greatly reduced organic compound like glucose. And if a chemolithoautotrope is using an electron donor and a higher redox potential than NAD plus NADP, they must use reverse electron flow to push the electrons back up the electron tower. And this is energetically unfavorable to the cell, consuming energy from the proton motive force to drive electrons in a reverse direction back through the electron transport chain. And some microbes are chemolithoheterotrophs, using an inorganic chemical for their energy and electron needs, but relying on organic chemicals in the environment for their carbon needs. And these organisms are also called mixotrophs, since they require both inorganic and chemical compounds for their growth and reproduction. So, talking about chemolithoautotropic bacteria, there are elemental sulfur granules present in the tissues of sulfur oxidizing bacteria that have been found from a submerged cave in central Florida. And most life on Earth depends either directly or indirectly on sunlight and photosynthesis to generate the organic carbon and cellular energy. However, the absence of light does not preclude life, and it's possible for diverse and complex communities to be fueled by microbial chemosynthesis. So, chemolithoautotropic bacteria are microbes basically that utilize chemicals from the bedrock. They use chemicals, that's why the chemo basically, and they utilize this from the bedrock, which is the litho, inorganic chemicals, as an energy source for making their own food. That's why autotroph. And the bacteria found in the caves that people are studying all around the world are chemolithoautotrophs, and they're utilizing sulfur from the bedrock to fuel their life cycle and possibly support the higher order crustaceans like crayfish, isopods, amphipods that are also found there. So while chemosynthetic microorganisms are found in nearly every environment on Earth, they're most abundant in habitats where darkness prevails and competition with photosynthetic organisms is eliminated. Some examples might be the hydrothermal vents and areas of the deep terrestrial subsurface like caves. Chemosynthetic bacteria can also contribute to the generation of biomass in caves. And in these nutrient poor habitats, the bacteria can fix CO2 into organic matter by the oxidation of reduced inorganic compounds such as iron, sulfur, and manganese, and actually provide a source of organic matter that fuels growth at higher trophic levels. And populations of chemolithotropic bacteria can be found in deep sea hydrothermal vents and often live in symbiotic relationships with other invertebrates. So, like for example, tube worms and clams, they live in association with chemolithotrophs at the bottom of the sea, and they have a very unique type of hemoglobin that provides the bacteria with a source of oxygen and hydrogen sulfide necessary for the chemoautotrophic metabolism. The bacteria in turn provides the worms bacteria provide the worms and clams with nutrition, since all the digestive system features are absent. It is estimated that these sulfide-based ecosystems in deep sea hydrothermal vents support over 500 different species of marine organisms. So yes, chemolithoautotropic bacteria is all the four options are true in case of this question, because basically these mean organisms that can obtain the necessary carbon for metabolic process from carbon dioxide in the environment. They can also use inorganic compounds such as nitrogen, iron, or sulfur for the energy to power these processes. So we can actually learn more about how different chemolithoautotropic bacteria get their energy. So this is chemolithotropy, as I talked about, the oxidation of inorganic chemicals for the generation of energy, and this process can use oxidative phosphorylation just like aerobic and anaerobic respiration, but here the substance being oxidized is an inorganic compound, and electrons are passed off to the carriers within the ETC or the electron transport chain, generating a proton motive force that is used to generate ATP with the help of ATP synthase. Moving on to the next question. Match the enumeration technique for cultured bacteria with the relevant parameter that is being studied. Anybody would like to try this? Anybody would like to try this before I explain? Okay, so the option will be 1, 2, that is qPCR will correspond with the number of target DNA RNA match basically with the number of target DNA RNA copies. Turbidity will match with optical density and color change of media and bubbles in Durham tubes will match with the most probable number. So we'll start with qPCR. qPCR is basically quantitative real-time PCR detection, which builds on the basic PCR of the polymerase chain reaction technique and allows researchers to estimate the quantity of starting material in a sample. And since the products are detected as the reaction proceeds, qPCR has a much wider dynamic range of analysis than conventional endpoint PCR. From a single copy to around 10 to the power 11 copies are detectable within a single run. And quantitative real-time PCR and subsequent amplicon detection is carried out in a closed tube format, which eliminates the need for post-PCR manipulations such as gel electrophoresis and significantly reduces the risk of cross-contamination. So what is the basic principle of qPCR? So when performing a qPCR, a fluorescent reporter dye is used as an indirect measure of the amount of nucleic acid present during each amplification cycle. The increase in the fluorescent signal is directly proportional to the quantity of the exponentially accumulating PCR product molecules or amplicons produced during the repeating phases of the reaction. And reporter molecules are categorized as double-stranded DNA binding dyes, dyes conjugated to primers, or additional dye conjugated oligonucleotides referred to as probes. So the use of double-stranded DNA binding dyes such as cybergreen 1 represents the simplest form of detection chemistry. So when free in solution or with only single-stranded DNA present, cybergreen 1 dye emits light at a very low signal intensity. As the PCR progresses and the quantity of double-stranded DNA increases, more dye binds to the amplicons and hence the signal intensity increases. Alternatively, a probe or contamination of 2 depending on the detection chemistry can add a level of detection specificity beyond the double-stranded DNA binding dye since it binds to a specific region of the template that is located between the primers. So the most commonly used probe format is the dual labeled probe. The dual labeled probe is an oligonucleotide with a 5' fluorescent label, for example 6-FAM, and a 3' quenching molecule, such as one of the dark quenchers, example BHQ or OQ. So these probes are designed to hybridize to the template between the 2 primers and are used in conjunction with the DNA polymerase that has inherent 5' to 3' exonuclease activity. So when the dual label probe is free in solution, the signal intensity is very low because the reporter dye is in close proximity to the quencher moiety. As more of the template is produced during the reaction, more probes hybridize to the template, which in turn are cleaved by the 5' to 3' exonuclease activity of the advancing DNA polymerase, and the fluorescent signal intensity increases as the 5' reporter dye is released into the solution. The use of probes labeled with different reporter dye allows for the simultaneous detection and quantification of the multiple targets in a single multiplex reaction. So a typical qPCR run consists of repeated cycles of alternating temperature incubation. Cycling procedure talks about the profile is often used when double-stranded DNA binding dyes such as molecular beacons or scorpion probes are the chosen detection chemistries. Primary extension is the most effective at 72 degrees Celsius because this is the optimal temperature for processivity of most DNA polymerases. At 72 degrees Celsius, polymerization occurs at a rate of approximately 100 bases per second. However, there is still processivity at lower temperature that is sufficient to amplify shorter templates, and qPCR amplicons are typically shorter than conventional PCR products. This extension is often combined with annealing in a single step at 16 Celsius when working with dual labeled probes. So how the cycling procedure occurs is at the beginning there is an initial denaturation. The reaction temperature is increased to 95 degrees Celsius and the sample is incubated for 2 to 10 minutes to ensure that all the complex targets are separated. Then there is a cycling setup which consists of denaturation, 95 degrees Celsius for 10 to 15 seconds to melt all the double-stranded DNA, basically to unwind the double-stranded DNA. Then there is the annealing. The temperature is lowered to 60 degrees for 30 seconds to promote primer and probe binding to the template. Then there is an extension. The subsequent elongation occurs at an increased temperature of 70 degrees Celsius, which is optimal for tagged DNA polymerase processivity. And this duration of this extension will be dependent upon amplicon size. And this cycle is repeated for 36 to 40 cycles. So this is the cycle procedure. This is the basic difference between PCR and qPCR. PCR is nothing but polymerase chain reaction. qPCR is a quantitative technique. Well, in real time you can actually see the number of amplicons, the number of copies. And there is primary amplification. In qPCR, either probe hydrolysis or fluorescence through intercalating dye. I already talked about this. PCR is non-fluorescence. qPCR is fluorescence because there is a real-time visualization readout. And these are the other gels. There are DNA bands on gel for PCR. You can actually see the bands on gel to see if your particular region of interest has been amplified or not. For qPCR, since it's fluorescence labeled, there is fluorescence involvement of fluorescence, you can actually see a real-time graph. qPCR is of high resolution amplification, PCR is compared to qPCR is of low resolution, and these are the applications. So the first question was qPCR, so that will match with number of target DNA and RNA copies, amplicons. Next is turbidity. Now what does turbidity mean? So these are three actually different photos. Okay, so what does turbidity mean, and how does turbidity how is it related to optical density? Optical density measurements of microbial and cell growth are one of the most common methods used in a microbiology lab. We also do that. And some of the main applications are the determination of the optimal time at which to harvest, the determination of the optical time to induce a culture when running a protein expression protocol, or the monitoring of cloning procedures. So the growth of cells, bacteria or yeast in liquid culture media is commonly controlled by measuring this optical density at 600 nanometer. It is also known as OD600 measurements, nanometer measurements. And OD600 measurements are typically used to determine the stage of growth of a bacterial culture. So these measurements help ensure that cells are harvested at a particular optimum time point that corresponds to an appropriate density of live cells. And since optical density is the case of in the case of OD600 measurements results from light scattering rather than light absorption, size and shape as well as dead cells and debris of cell may add to light dissipating. So distinctive cell types that are at densities of the same level may therefore show varying values of OD600 when estimated on a similar instrument. Growth of bacterial cells typically progresses through a series of consecutive phases including lag, log, stationary and decline. In general, cells should be harvested towards the end of the log phase using the optical density of the samples to determine when this point has been reached. Cells are routinely grown until the absorption or the absorbance at 600 nanometer reaches approximately 0.4 prior to induction or harvesting. So that is what is known as optical density. Now we'll move on to the Beer-Lambert's law. So we all know the Beer-Lambert law, which is basically empirically relates the absorption of light to the properties of the sample. So this law states that there is a logarithmic relation between the transmission of light through a specific sample, which is called the T. Just T is I by I0, which I is the outgoing light and I0 is the incident light. And there is a molar extension coefficient for a specific compound, which is the E, epsilon basically. The concentration of the absorbing species in the material, which is denoted by C, and the distance that the light travels, which is denoted by D. So OD estimations have gotten synonymous with estimations of bacterial concentration or number as per this Beer-Lambert law. However, measurements of OD are actually measurements of turbidity. So we can therefore apply the Beer-Lambert's law with certain contemplations just for microbial colonies of low densities. So for turbid samples such as cell culture, the major contributor for the absorbance measured is light scattering and not the result of the molecular absorption following the Beer-Lambert law. The measurements are therefore depending on the optical setup of the spectrophotometer. Different instrument types will most likely tend to give different results, different OD600 readings for the same turbid sample. Therefore, if results from different spectrophotometers are to be compared, they must be normalized first by either a simple correlation of the OD readings using the same sample on two different instruments to compare, or with a more precise approach by the creation of an appropriate calibration curve. A calibration curve can be constructed by comparing the measured OD600 to expected OD600 over a range of different concentration, and expected OD600 is determined by counting the cell number using an alternative technique and converting to OD600 using the rule of thumb that 1 OD600 is equal to 5 into 10 to the power 8 cells per ml for E. coli. So the use of disposable cuvettes is recommended rather than micro nano volume technologies for optical density measurements of cell solution culture solutions. The amount of cells is reflected in the reading, and the likelihood of the fluctuating amount of cells in a drop form from sample to sample can be considered as extremely significant, and it's therefore recommended to use cuvettes since the amount of error in a bigger volume is not as significant, and the cuvette measurements provide a bigger average and therefore more reproducible readings. Also, to prevent the suspension settling too quickly and giving an OD reading that changes with time, glycerol should be added to the sample. So this is how OD relates to optical density relates to turbidity. Then moving on to the most probable number or the color change in the habitates. So the most probable number analysis is basically a statistical method based on the random dispersion of microorganisms per volume in a given sample. So in this method, measured volumes of water are added to a series of tubes containing a liquid indicator growth medium. The media receiving one or more indicator bacteria show growth and a characteristic color change. The color change is absent in those receiving only an inoculum of water without any indicator bacteria. And from the number and distribution of positive and negative reactions, the most probable number of the indicator organisms in the sample may be estimated by reference to statistical tables. At this entire test, the MPN test is conducted in three steps: the presumptive test, the confirmed test, and the completed test. So, what happens in the presumptive test? This test specifically is a specific enrichment procedure for coliform bacteria and is conducted in fermentation tubes filled with a selective growth medium, such as McConkey lactose growth, which contains inverted Durhan tubes for the detection of fermentation gas. A series of lactose growth tubes are inoculated with measured amounts of the water sample to be tested. For example, the series of tubes may consist of 3 or 4 groups of 3 or 4, 5 more tubes, and the main selective factors found in the medium are lactose, sometimes surfactants such as sodium lauryl sulfate or a sodium taurocolate, and often a pH indicator dye for facilitating detection of acid production, such as bromkisol purple or brilliant green. And the selective action of lactose occurs because many bacteria cannot ferment this sugar, whereas coliform bacteria and several other bacterial types can ferment it. The surfactant and dye do not inhibit coliform bacteria, whereas many other bacteria, such as the spore formers, are inhibited. Then the second step is the confirmed test. This test serves to confirm the presence of coliform bacteria when either a positive or doubtful presumptive test is obtained. A loopful of growth from such a presumptive tube is transferred into a tube of brilliant green lactose bile, 2% broth, and incubated at 35 degrees Celsius for 48 hours. This is selective medium for collecting coliform bacteria in water, dairy, and other food products. A selective agent in the medium is lactose. The broth tube also contains a Durham tube to detect gas production basically. And then there's a plate of less endo agar or EMB agar, which is streaked with a loopful of growth from a positive tube and inoculated for 18 to 24 hours at 35 degrees Celsius. And typical coliform bacteria like E. coli and Enterobacter aerogenes, they exhibit good growth on this medium and form red to black colonies with dark centers on a sheen. Salmonella typhi exhibit a good growth, but the colonies are colorless, and Salmonella aureus growth is inhibited altogether. The third step is the completed step. This step helps to further confirm doubtful and if desired positive confirmed test results. A typical coliform colony from an LES endo-agar plate is inoculated into a tube of brilliant green bile broth and on the surface of a nutrient agar gland, and they are then incubated for 24 hours at 35 degrees Celsius. And after 24 hours, the broth is checked for the production of gas and a gram stain is made from organisms on the nutrient agar gland. If the organism is gram negative, non-spore forming rod and produces gas in the lactose tube, then it is positive that coliforms are present in the water sample. So this is how the broth looks like, and this is the Durham tube. You can see the gas production leads to a bubble in the Durham tube. So the main objective of this most probable number experiment is to enumerate the number of bacteria present in the drinking water by the MPN method and to identify the bacteria present in the drinking water sample. So this is how the entire thing works out. There are 3 steps to identify each kind of bacteria. So this is how the Durham tubes show gas formation if there is any kind of gas formation with the presence of the indicator bacteria. So this is why this question, color change of media and bubbles in Durham tubes, will match with the most probable number experiment. So that is how this entire match and pair can be solved. Okay, moving on to the next question. Consider the following steps for microbial community fingerprinting. Anybody? What can be the following steps for microbial, what can be the step-by-step procedure for microbial community fingerprinting? Anybody? So it will be, the first step will be to extract the DNA. Obviously, if you are working with the DNA, the first step will be isolating the DNA from the organism or from the tissue sample or the since it's microbial community, basically organism. then you have to do the PCR of the gene of interest. So if you have a particular gene of interest, we'll actually need to amplify that before going on with the further steps. So first of all, we'll do the polymerase chain reaction to amplify the gene of interest. Then we'll separate and purify our PCR product or our gene of interest amplified version, and then we'll send it for DNA sequencing. So this is how a microbial community fingerprinting based on DNA sequencing can occur. Okay, moving on to the next question. To add bacterial strains in a contaminated site to accelerate the remediation is known as. Okay, so it's known as bioaugmentation, and this is how bioaugmentation basically pans out. It's basically the addition of microorganisms that have the ability to degrade recalcitrant molecules in the polluted environment, and this approach is much less costly because you're actually using organisms in the natural environment, and also friendlier because these are natural organisms that are you're adding to the environment compared to the physical chemical approaches or chemical approaches that you take. That's much harsher to the environments compared to the bioaugmentation, which is done by the microorganisms which are already present in the environment. It's a very clever way of biodegrading recalcitrant molecules from our polluted environment. So the answer is bioaugmentation. And what is the difference between bioaugmentation and biostimulation? So this is a table that talks about exactly this. Bioaugmentation is the process of adding specific microorganisms to enhance the existing populations and promote the biodegradation process, whereas biostimulation is the process of adding electron acceptors, electron donors or nutrients to stimulate naturally occurring microbial populations in the contaminated area. So the end point objective of both are kind of almost the same. In case of bioaugmentation, you actually put bacteria directly onto that place where you want bioremediation to happen. In case of biostimulation, you add electron acceptors, electron donors, and various specific nutrients to that particular place, and you stimulate those microbial populations to come out naturally. You're not adding any microbial population to the place. You're adding the nutrients responsible for powering that microbial population, and that is how you're doing biostimulation. So biostimulation is indigenous microorganisms, obviously, because you're only adding the nutrients artificially. Everything else is happening there only. It's very indigenous. For bioaugmentation, it's exogenous microorganisms because you're adding microorganisms from a different environment, maybe from a different ecosystem to it to help in the bioremediation process. Bioaugmentation basically occurs with cultured microorganisms because you're obviously taking lab microorganisms, you're culturing the microorganisms. You're not randomly taking microorganisms from one place and adding it to the other. You're taking cultured microorganisms which have spent time in the lab, and then you're putting it in the site of interest for bioaugmentation to occur. However, biostimulation, the addition is of only nutrients and electron acceptors. Everything else will happen naturally. Everything else will occur naturally. The bioaugmentation in bioaugmentation, the introduced microbe often cannot be established in the environment, and these introduced organisms rarely survive in the new environment. So you're taking your lab culture strains and you're introducing those to the new environment, new wildlife environment, and sometimes those microbes do not really have it in themselves to survive in such a wild condition. So then they pan out and they die. But in biostimulation, the drawback in biostimulation is that due to contaminant toxicity, the existing microbial population may not be enough for the bioremediation process. So you're actually adding nutrients and everything etc. And you're actually stimulating the formation of natural populations. But since there's already a lot of contamination in that particular area, the existing microbial population may not survive enough for the biodegradation process to occur. So these are some of the drawbacks of both the processes. Moving on to the next question. Some heavy metals can be transformed into less toxic or less mobile version by changing their redox state, and this is known as transformation. Any compound that is unfamiliar to the environmental microbes can be referred to as a xenobiotic. Anybody? Anybody would like to answer? like to try at least? Okay, so it's true. Both of them are independently very true statements. So before moving on to xenobiotics, I'll tell you what happens in case of heavy metals. So heavy metals are very persistent in nature and are very toxic to every life form that is there. Increase in industrialization, urbanization, and unsafe agricultural practices is constantly adding heavy metals to the environment and consequently causing heavy metal pollution of water and soil. Considering the negative impacts of heavy metals in the environment, several strategies have been devised to remediate them. However, most of these have their own limitations as well. Bioremediation of metals by microorganisms is efficient, cost effective, and environment friendly method of metal detoxification. And microbes can utilize metal contaminants as their energy source and transform them to less toxic forms. When exposed to metals for a considerable period of time, microorganisms interact with them and become tolerant by developing resistant mechanism against them. So metal microbe interactions can occur in several ways such as biosorption, bioleaching, biomineralization, bioaccumulation, and biotransformation. And study of these interactions is important to understand resistant mechanisms against metals, which include barriers, efflux systems, sequestration, and reduction of metals. So these mechanisms are encoded by the resistance genes localized in the chromosomes or the plasmids. And understanding these resistant mechanisms against metals in microorganisms becomes very crucial for devising strategies for bioremediation of metals. So this part is true. So what is xenobiotic? A xenobiotic is basically a chemical substance found within an organism that is not naturally produced or expected to be present within the organism. So it can also cover substances that are present in much higher concentrations than are usual. So natural compounds can also become xenobiotics if they are taken up by another organism, such as the uptake of natural human hormones by fish found downstream of sewage treatment plant outfalls, or the chemical defenses produced by some organisms as protection against predators. So xenobiotic, this term is used to describe chemical substances that are foreign to animal life and thus include examples such as plant constituents, drugs, pesticides, cosmetics, flavorings, fragrances, food additives, industrial chemicals, and environmental pollutants. So this is what xenobiotics is all about. So that statement is also true. Any compound that is unfamiliar to the environment, to environmental microbes, can be referred to as xenobiotic. So the next question, polybeta hydroxybutyrate and polybeta hydroxyvalidate are microbially produced and serve as a form of energy storage molecule to be metabolized when other common energy sources are not available. In denaturing gradient chain electrophoresis, the DNA that has more GC bonds travels slower. This can be due to the guanine and cytosine that are weaker than adenine and thymine. Which statement do you think is true? Which of the options is true? Anybody? At least try any one statement. If you want to comment on any one statement, that's also fine. At least try. Trying is the most important. Anybody? Anybody? Giving you time to think. So statement A is true, statement B is false. So I'll talk about this. Polyhydroxybutirates are basically members from family of the polyesters known as polyhydroxyalkanoids. This slide has been taken from the lecture videos only, and they accumulate in intracellular granules by gram-positive and gram-negative microorganisms. These are produced when there is excess carbon source with the limitation of one of the essential nutrients. These are also known as biopolymers, as they are produced from microorganisms, and they are thermoplastic polymers and totally biodegradable. So the first option is very much true. These are microbially produced, and they do serve as a form of energy storage molecule to be metabolized when other common energy sources are not available. So these are synthesized as intracellular storage material and accumulates as distinct white granules during unbalanced growth in the cell, and these are also clearly visible in the cytoplasm of the cell. Many bacteria, including those in the soil, are capable of polyhydroxybutyrate production and breakdown. And what is the significance of these polyhydroxybutyrate and why are they produced? They are polymers that bacteria produce under conditions of low concentrations of important nutrients, typically nitrogen, but sometimes oxygen as well, and high concentrations of carbon sources. And this process occurs because the excess carbon leads to bacteria creating carbon reserves to save for a time with more plentiful nutrients in which they need energy to carry out regular functions. So bacteria store PHBs in granules for later use, and these polymers are accumulated intracellularly under conditions of nutrient stress and act as a carbon and energy reserve. So these slides have been taken from the lecture videos only. I know these are a little bit blurry, but you can actually go back to the lecture videos and read these slides. Next, moving on to the second option, which was false, we'll talk about denaturing gradient gel electrophoresis. We all know the basics of normal gel electrophoresis, but then there are a lot of modifications that have been made to the system to actually help scientists do science basically. So what is gradient gel electrophoresis and what does it help to separate, and what is the principle? So we know that DNA is responsible for preserving genetic information across species and time, that we know, and mutations can occur randomly or induced as an organism adapts to new environments or its surrounding. So observing these genetic changes can be a very difficult task, and this is where DGGE or denaturing gradient gel electrophoresis comes in to assist by analyzing these mutations in a certain population. So the basic principle is to separate DNA strands based on the ratio of GC and AT base pairs. During the process, the DNA is exposed to a gradient of denaturant at high temperatures within a polyacrylamide gel. As the DNA sample progresses through the gel from low to high denaturant concentration, it begins to melt at varying points. The theory goes that at higher GC content makes it harder to melt, while samples with lower GC content tend to melt more rapidly. So denaturing gradient gel electrophoresis is basically a technique used for separating DNA fragments according to their mobilities under increasingly denaturing conditions, usually increasing formamide or urea conditions. Small samples of DNA are added to an electrophoresis gel that contains a denaturing agent. The denaturing gel induces melting of the DNA at various stages. As a result of this melting, the DNA spreads through the gel and can be analyzed for single components. DGG analysis are employed for the separation of double-stranded DNA fragments that are identical in length but different in sequence. So in practice, the DNA fragments are usually produced via PCR amplification. The DGG technique exploits, among other factors, its difference in the stability of the GC pairing, which has 3 hydrogen bonds per pairing, as opposed to the AT pairing, which has 2 hydrogen bonds. A mixture of DNA fragments of different sequence is separated by electrophoresis on an acrylamide gel containing a linearly increasing gradient of DNA denaturants, usually urea or formamide. In general, the DNA fragments richer in GC will be more stable. GC is guanine and cytosine triple bonds. They will be more stable and remain double-stranded until reaching higher denaturant concentrations. Double-stranded DNA fragments will migrate better in the acrylamide gel, while denatured DNA molecules slow down or stop in the gel. In this manner, DNA fragments of different sequence can be separated in acrylamide gel. So this technique is commonly performed for partial 16S rRNA gene, but also functional genes may be used. A GC-rich sequence can be incorporated into one of the primers used in the PCR to modify the melting behavior of the fragment of interest and to improve the separation of the fragments. So the DGG gels can be stained with DNA-binding fluorescent dyes such as cyber green and visualized under UV light. Known standards may be used for comparing the samples on different gels. Ideally, one band on the gel corresponds to one species, and therefore the number of bands gives an idea of the diversity of the sample. The gene fragments can be excised from the gel, eluted, that is into sterile water, and amplified for sequencing. The relative abundance of various microorganisms can be estimated by measuring the intensity of the bands relative to the intensity of all bands in the corresponding sample. So, what are the advantages? It's very sensitive to variations in DNA sequence, allows simultaneous analysis of multiple samples. It is a useful method for monitoring shifts in the community structure over time. Community profiles can be analyzed with cluster analysis. The use of universal primers allows the analysis of microbial communities without any prior knowledge of the species. And the fragments separated by DGG can be excised, cloned, and sequenced for identification. And it's possible to identify constituents that represent only 1% of the total community. So DGG can be applied to phylogenetic and functional genes. However, there are also some disadvantages of this process. This type of analysis is rather time consuming. It analysis suffers community analysis same drawbacks such as all PCR based community analysis techniques, including biases from DNA extraction and amplification. The variation in the 16S RNA gene copy number in different microbes make this technique only semi-quantitative. Then there's microheterogeneity in RNA encoding genes present in some species that may result in multiple bands for a single species and subsequently to an overestimation of community diversity. Then heteroduplexes can cause biases to the observed diversity. No method for automated analysis currently available. It works well only with short fragments, that is less than 600 pp, thus limiting phylogenetic characterization. Gels of complex communities may look smeared due to the large number of bands. Band position does not provide reproducible taxonomic information, and its like results are very difficult to reproduce between gels and labs. DNA-based DGG is a monitoring tool. It is especially suitable for samples with few species and gives complementary information on species diversity when combined with other methods. And it is also very well suited for analyzing community changes over time. Very suitable technique for the identification of novel or unknown organisms. The detection sensitivity is limited due to PCR and primer artifacts. Usually, the most abundant species are detected, but it's easy to perform. The most abundant species can be readily detected, and limited information about the abundance of detected species and no information about the activity of the detected species. So, long analysis time that is almost covering spanning days, several days, can make it very difficult to use as an online tool. So that is what a denaturing gel electrophoresis technique is all about. Moving on to the next question. Aromatic contaminants are difficult to remediate because these are the four options. Which one would you select as the correct option? Anybody? Anybody would like to try? Anybody? Anybody would like to try? Okay, so the reason aromatic contaminants are very difficult to degrade or remediate is because they have a very stable electron resonance happening in the ring, because they have the aromatic ring, so there is always resonance that is happening. So, what are aromatic contaminants basically? Aromatic contaminants are very similar to naphthenes, but they contain a resonance stabilized unsaturated ring core, and they are compounds that contain at least one benzene ring. And the benzene ring is very stable and does not crack to smaller components. So, aromatics are not a preferred feedstock because few of the molecules will never crack. Because few of the molecules will crack and the most will never crack. The cracking of aromatics mainly involves breaking of the side chains, resulting in excess fuel gas yield. In addition, some of the aromatic compounds contain several rings, which are called polynuclear aromatics (PNAs), that can compact to form what is commonly called as chicken wire. So, basically, some of these compound aromatics will end up on the catalyst as carbon residue, and some will become slurry product. In comparison, the cracking paraffins, cracking aromatic stocks results in lower conversion, lower gasoline yield, and less liquid volume gain, but with higher gasoline octane. So, aromatic compounds are both common growth substrates for microorganisms and prominent environmental pollutants. So, the crucial step in the degradation is overcoming the resonance energy that stabilizes their entire ring structure. So they are, because they are unsaturated hydrocarbons which have one or more planar six carbon rings called benzene rings, to which hydrogen atoms are attached. And many aromatic hydrocarbons contain a benzene ring, which is stabilized by resonance, and the pi electrons are delocalized in the ring structure. So it's basically a class of organic aromatic organic molecule, and these are very carcinogenic in nature, very extremely hydrophobic in nature, and resistant to biological degradation. Extraction of these polycyclic aromatic hydrocarbons from environmental matrices is the first and most crucial step in this quantification. Extraction followed by quantification is essential to understand the extent of contamination prior to the application of remediation approach. So, due to their non-polar structures, these can be adsorbed tightly to the organic matter in soils and sediments, making them more difficult to be extracted. So, extraction can be achieved by a variety of methods. Techniques such as supercritical and subcritical fluid extraction, microwave assisted solvent extraction, plant oil assisted extraction, and some micro extraction techniques provide faster polycyclic aromatic hydrocarbon extraction using less organic solvents, while providing a more environmentally friendly and safer process with minimum matrix interferences. And more recently, more environmentally friendly methods for soil and sediment remediation have been explored, and this often involves using natural chemicals such as biosurfactants to solubilize this in contaminated soils and sediments to allow subsequent microbial degradation. So this is how quantification occurs, and after that the degradation process is carried on. But the main reason why it's so difficult to push these polycyclic aromatic hydrocarbons for bioremediation is because they have this pi electron resonance thing happening because of the ring structure that's very difficult to crack, very difficult to remediate. It's a very stable structure, but they are very like they are villains to the society because they are very carcinogenic micropollutants which are resistant to environmental degradation due to their highly hydrophobic nature, and concerns over their adverse health effects have resulted in extensive studies about the remediation of soils contaminated with these polycyclic aromatic hydrocarbons. Moving on to the next question. Microbes need acclimation time because these are the four options. Which one do you think is the correct? Anybody? Still in the meeting, right? So now, Fleming, anybody would like to try? So the answer is the fourth option, new environmental conditions and low initial numbers. I think it's very self-explanatory. Everyone needs acclimation time, not only microbes. If even we go to a new place, we need some time to actually adapt to the environment and acclimatize to the environment, to the situation, to everything around us. So that's why acclimation time is needed. For microbes, however, the reason is low initial numbers. They have very low initial numbers because they'll take time to come from a different environment and actually be comfortable in the new environment, so that they can actually amplify their numbers. So acclimatization or acclimation is the process in which an individual organism adjusts to the change in the environment, such as change in altitude or temperature or humidity or pH, anything, allowing it to maintain fitness across a range of environmental conditions. It occurs in a short period of time and within the organism's lifetime, and this may be a discrete occurrence. For example, when mountaineers acclimatize to high altitude over hours or days, or may instead represent part of a periodic cycle, such as a mammal shedding heavy winter fur in favor of a lighter summer coat. So, organisms can adjust their morphological, behavioral, physical, or biochemical traits in response to the changes in the environment. And while the capacity to acclimate to novel environments has been well documented in thousands of species, researchers still know very little about how and why organisms acclimate the way that they do. So, there is the biochemical need in order to maintain performance across a range of environmental conditions. There are several strategies organisms use to acclimate. In response to changes in the temperature, organisms can change the biochemistry of cell membranes, making them more fluid in cold temperatures and less fluid in warm temperatures by increasing the number of membrane proteins. In response to certain stressors, some organisms express so called heat shock proteins that act as molecular chaperones and reduce denaturation by guiding the folding and the refolding of proteins. It has also been shown that organisms which are acclimated to high or low temperatures display relatively high resting levels of heat shock proteins, so that when they are exposed to even more extreme temperatures, the proteins are readily available. Also, expression of heat shock proteins and regulation of membrane fluidity are just two of the many biochemical methods organisms use to acclimate to novel environments. Then there is this morphological aspect. Organisms are able to change several characteristics relating to their morphology in order to maintain performance in novel environments. For example, birds often increase their organ size to increase their metabolism. This can take the form of an increase in the mass of nutritional organs or heat producing organs, like the pectorals, with the latter being more consistent across species. So, while the capacity of acclimatization has been documented in thousands of species, there's still studies going on. And since researchers began to first study acclimation, the overwhelming hypothesis has been that all acclimation serves to enhance the performance of the organism. And this idea has come to be known as the beneficial acclimation hypothesis. And despite such widespread support for the beneficial acclimation hypothesis, not all studies show that acclimation always serves to enhance performances. One of the major objections to this beneficial acclimation hypothesis is that it assumes that there are no costs associated with acclimation. However, there are likely to be costs very much associated with acclimation. These include the cost of sensing the environmental conditions and regulating responses, producing structures required for plasticity, such as the energetic cost in expressing heat shock proteins, and genetic costs such as linkage of plasticity chains with harmful genes. So, given the shortcomings of the beneficial acclimation hypothesis, researchers are continuing to search for a theory that will be supported by empirical data. And the degree to which organisms are able to acclimate is dictated by their phenotypic plasticity, or the ability of an organism to change certain traits. And recent research in the area of acclimation capacity has focused more heavily on the evolution of phenotypic plasticity rather than acclimation responses. Scientists often believe that when they understand more about how organisms evolve the capacity to acclimate, they will better understand acclimation. So the correct answer is the fourth option. Moving on to the next question. Commensal bacteria are only found in biofilms. Is it true or false? What do you think? And then solar flaming. Do try what you know the meaning of commensalism. So, do you think commensal bacteria can only be present in the biofilms? Ready? So it's false basically, and this is what commensalism basically means. It's a symbiotic relationship where one organism benefits without harming the other. So it refers to the analogy that says that eat from the same dish. In this kind of relationship, neither benefits from the other, nor does it provide any kind of harm. It is therefore a neutral relationship. Uh, like one of the organism is benefiting from the other, but the other organism is not benefiting, neither it is being harmed. So there are other classes of relationships such as mutualism, in which both organisms obtain benefits, or parasitism, in which one profits from the other by harming it. Although friendly bacteria inhabiting our organism are usually referred to as commensal, the research in this field suggests that the relationship between our gut microbiota and us is not merely commensal, but rather a mutualistic relationship. So this is what commensalism means. You can see from this sketch over here what commensalism basically means. One organism is this is mutualism, where both the organisms mutually benefit from each other. So the bee is taking the honey, and the pollen of the flower is scattered everywhere by the bee. So that is how both are, it's a mutualistic affair. There is commensalism, where one species is getting benefited from this species, but this species is neither getting harmed by this nor getting benefited. So the entire thing is zero, neutral. Parasitism is mosquitoes, how they suck blood from human beings. So it's getting, the mosquito is benefiting from the human, but the human is getting it's losing blood. So it's kind of a negative impact that's happening. It's harming the human. That's how it's a parasitism. So some differences between mutualism and commensalism. So I already talked about how these relationships are very different, and some of the examples are also shown here. You can always go online and find more examples. So the main question was, does commensal bacteria are only found in biofilms or not? So that's false. Commensal bacteria are actually found in a lot of places. Sorry, this slide is a little messed up. So this is how throughout the human body also you can find commensal bacteria, and how their localization is restricted to some particular regions. So a diverse community of trillions of commensal bacteria actually inhabits mucosal and epidermal surfaces in humans, and they play an important role in defense against pathogens, including respiratory pathogens. And commensal bacteria act on the host's immune system to induce protective responses that prevent colonization and invasion by foreign pathogens. On the other hand, these bacteria can directly inhibit the growth of respiratory pathogens by producing antimicrobial products, signals, and competing for nutrients and adhesion sites. Such mechanisms preserve the niche for commensal bacteria and support the host in containing respiratory infections. So there are a lot of papers that discuss about the role of commensal bacteria in conferring protection against respiratory pathogens and the underlying mechanisms by which these bacteria do so. and a deeper knowledge of how commensal bacteria might provide new insights that are poised to aid in the development of vaccines and therapeutics to target infectious diseases. So commensal bacteria actually produce protection in mouse models, also in humans. We'll discuss a little bit about the humans only. So it's very little information is available whether commensal bacteria can prevent respiratory infections in humans. Oral commensals such as Streptococcus oralis and Streptococcus salivarius can actually induce protection against middle ear inflammation, referred to as otitis media, which is primarily caused by respiratory pathogens such as S. pneumoniae and Haemophilus influenzae. So, upon intranasal administration of S. salivarius and S. oralis, children susceptible to acute otitis media displayed reduced recurrence of disease with no side effects. And contrarily, a nasal spray containing oral commensals, example S. vitis and S. oralis, in susceptible children under 4 years of age even, showed no significant effect regarding the number of episodes of recurrent otitis media compared to the placebo group. So the discrepancy in these studies might be due to the differences in bacterial doses, inoculation regimens, and combinations, which need to be analyzed in light of the new technologies such as metagenomics and next generation sequencing and concepts like dysbiosis. So the fact that antibiotics were used together with the streptococcal nasal spray is the first study but not in the second is also an important factor to consider. In controlled infection studies in human, nasopharyngeal colonization by the commensal Neisseria lactamica provided protection against the respiratory pathogen Neisseria meningitis. And furthermore, in a block randomized challenge trial, 310 healthy individuals were intranasally inoculated with live Neisseria lactamica or sham, and the bacterial carriage was monitored for 26 weeks. And all those who developed nasopharyngeal colonization by N. lactamica revealed a significant reduction in the N. meningitis carriage compared with sham treated ones. So these studies show that commensal bacteria not only show inhibitory or displacing effects on the carriage of respiratory pathogens, but also highlight the ease and safety with which these bacteria can be used to contain infections in humans. It is, however, notable that most bacteria with high pathogenic potential, such as those in the examples, are also part of the healthy microbiome. So the reason as to why these pathogens cause diseases is attributed to various host and microbial factors, including viral infections. Like dysbiosis in particular, such as a result of antibiotic use, has been associated with a reduction in the prevalence of respiratory commensal bacteria like Corynebacterium species and others in the nasopharynx of healthy infants. So these are considered to reduce the colonization by S. pneumoniae and Haemophilus influenzae S. aureus in the respiratory tract. It is further shown that respiratory syncytial virus infection in children below 2 years of age was positively correlated with nasopharyngeal Haemophilus influenzae and Streptococcus microbiota clusters, and inversely correlated with Staphylococcus aureus. And then there was a transcriptomic study conducted in children infected with Haemophilus influenzae and Streptococcus clusters, which presented greater expression of immune components, suggesting that nasopharyngeal microbiota can actually influence host immunity. And in line with this, prolonged antibiotic treatment in early life has also been annexed with an increased risk for respiratory infections in infants. Thus, all these studies shine light on the effect of the microbiota perturbations caused by antibiotics on host susceptibility to respiratory infections, particularly during the critical life period of immune maturation. So there's a lot of ways how commensal bacteria actually mediate protection in humans. So the question was, commensal bacteria are only present in biofilms, which is definitely not true. They are present in a lot of places, including human microbiota. So that's how this question can be answered. Moving on to the next question. Gut health is linked with gut microbiome. Is it true or false? Anybody? Try. Anybody? Anybody? So the answer is true. Definitely, gut health is definitely linked with gut microbiome biome. So trillions of bacteria, as we know, are living on our skin and in our body, and in fact, there are likely more bacterial cells in your body than human cells, and that's fascinating. And it's estimated that in a 154 pound or 70 kg man, there are around 40 trillion bacterial cells and only 30 trillion human cells. So, and most of these bacteria live in a part of your large intestine called the cecum. And there are hundreds of different types of bacteria in the intestines. While some may cause disease, most of them carry essential tasks to keep you healthy. For example, your gut bacteria produces certain vitamins, including vitamin K, and communicate to the immune system to help your body fight off infection. They also influence how you digest certain foods and produce chemicals that help make you feel full. So therefore, your gut bacteria may influence your weight as well. So they affect how your food is digested. So since your gut bacteria line the, line your intestines, they come into contact with the food you eat. So this may affect what nutrients you absorb and how energy is stored in your body. So one study examined the bacteria in 77 pair of twins, one of whom had obesity and one of whom did not. And like like this study found that those who had obesity had different gut bacteria than their twins without obesity, and particular obesity was associated with lower gut bacteria diversity, meaning there were fewer types of bacteria in the gut. And other studies have shown that if the gut bacteria from people with obesity are put into mice, the mice gain weight. So this suggests that the gut bacteria can easily affect weight as well. So this may be due to the effect of bacteria on the digestion of different types of foods. For example, humans can't digest fiber, but certain gut bacteria can. But digesting fiber, these gut bacteria produce a number of chemicals that benefit gut health and possibly promote weight loss. For example, a number of studies have shown that people with high fiber intake have a lower weight, which may be due to the role that gut bacteria play in digesting fiber. A recent study found that the ratio of two types of bacteria in your intestines may determine how much weight you lose when given a particular diet. These two bacteria are Revotella, which digests fiber and carbohydrates, and Bacteroidetes, if I'm not wrong, which people who eat more animal protein and fat have more of. So in this study, 62 people were given a high fiber whole grain diet for 26 weeks. Those who had more Revotella in their intestines lost 2.3 kg more body fat than those with more bacteroidetes in their intestines. So your gut bacteria can digest certain antioxidants found in plants known as flavonoids, which may also help prevent weight gain. So finally, your gut bacteria can influence how dietary fats are absorbed in the intestines, which may affect how fat is stored in the body. So this is how gut bacteria can affect your bodily weight. Basically, it can also affect inflammation. So inflammation occurs when your body activates your immune system to fight against infections by foreign pathogens. So it can also be caused by an unhealthy diet. For example, a diet containing too much fat or sugar or calories can lead to elevated inflammatory chemicals in the bloodstream and fat tissue, which may contribute to weight gain. Your gut bacteria play an important role in inflammation. Some species produce chemicals like lipopolysaccharides which cause inflammation when they pass into the blood. When mice were given this kind of lipopolysaccharides, they gain as much weight and have similar increase in blood sugar and insulin as mice fed with a high fat diet. So therefore, certain gut bacteria that produce LPS and cause inflammation may contribute to the weight gain and insulin resistance. So there was a study wherein it was conducted in almost 300 people to 300 people, and it was found that those who were overweight had lower gut bacteria diversity and higher levels of C-reactive protein, which is an inflammatory marker in the blood. However, some species of the intestinal bacteria can reduce inflammation and prevent weight gain as also. So, the names are Akkermansia and Bifidobacteria. These are some of the beneficial species of bacteria that help maintain a healthy gut barrier and prevent inflammatory chemicals from passing from the gut into the bloodstream. And studies in mice have found that Akkermansia can reduce weight gain and insulin resistance by reducing inflammation. Similarly, when mice were fed prebiotic fibers to help increase bifidobacteria in the gut, weight gain and insulin resistance decrease without affecting energy intake. So this is a relatively new area of research. Therefore, it's still not clear how gut bacteria affect inflammation and weight in humans. Also, they produce chemicals that help you feel hungry or full. So, your body produces a number of different hormones that legit affect your appetite, including leptin, ghrelin, peptide, etc. Some studies have shown that different bacteria in the gut can affect how much of these hormones are produced and whether you feel hungry or full. So, short chain fatty acids are chemicals that are produced within when certain species of gut bacteria break down fiber. One of these is known as propionate. And one study in 60 overweight adults found that taking propionate for 24 weeks significantly increased levels of hormones PYY and GLP-1, both of which influence hunger. People who took propionate also had reduced food intake and reduced weight gain. Other studies have shown that prebiotic supplements, which contain compounds that are fermented by gut bacteria, can have a similar effect on appetite. People who ate 16 grams of prebiotics per day for 2 weeks had higher levels of hydrogen in their breath, and this indicates gut bacterial fermentation, less hunger, and higher levels of the hormones GLP-1 and PYY, which make you feel full. So, the bacterial cells harbored within the human gastrointestinal tract or the gut outnumber the host cells by a factor of 10, and the genes encoded by the bacterial resistant within the gut outnumber their host genes by more than 100 times. So these human digestive tract associated microbes, these are referred to as the gut microbiome, and the human gut microbiome has a very huge role in both health and disease, and it has been a subject of extensive research establishing its involvement in human metabolism, physiology, nutrition, immune function, etc. An imbalance of the normal gut microbiota have been linked with gastrointestinal conditions such as inflammatory bowel disease or IBD, in irritable bowel syndrome or IBS, and wider systematic manifestations of diseases such as obesity, type 2 diabetes, and atopic. So there are various reviews and papers online you can search where they evaluate their evolving knowledge of the development, complexity and functionality of the healthy gut microbiota, and the ways in which the microbial community is perturbed in dysbiotic disease states. So the gut microbiota is mainly composed of strict anaerobes, which outnumber the facultative anaerobes, organisms able to grow both aerobically and anaerobically, and also the aerobes by almost 100-fold. So, although the presence of more than 50 bacterial phyla has been detected in the human gut today, the microbiota are dominated by only two phyla, the Bacteroidetes and the Firmicutes. I've already discussed about these. And estimates of the number of bacterial species present in the human gut vary widely among species. Also studies, but it is generally accepted that individuals harbor more than 1,000 microbial species level phenotypes. So the gut has a lot of functions, including metabolism, because they encode a larger number of proteins. There is a lot of function that they have in host protection and immune system development, because many intestinal bacteria produce antimicrobial compounds and compete for nutrients and sites of attachment in the gut lining, thereby preventing colonization by pathogens. So this action is known as the barrier or competitive exclusion effect. Host cells in the gut wall have attachment sites that can be used by pathogenic bacteria to enter the epithelial cells. And in lab studies, it has been seen that non-pathogenic bacteria can be seen to compete for these attachment sites in the border of the intestinal epithelial cells, preventing the attachment and subsequent entry of pathogenic enteroinvasive bacteria into the epithelial cells. Furthermore, because bacteria compete for nutrients in their immediate surroundings and maintain their collective habitat by administering and consuming all resources, the enteric microbiota can outcompete pathogenic bacteria for resources by sheer force of numbers. In addition, bacteria can inhibit the growth of their competitors by producing antimicrobial substances such as bacteriocins, and the ability to synthesize these bacteriocins is widely distributed in your gut gastrointestinal bacteria. So this is how the gut microbiome basically is involved in a lot of health. Basically uh, is involved in the health and disease state of your body. So as you can see from the schematic also present on the screen, that these are the different gut microbiota stains that you can see in stomach, duodenum, gut brain axis, gut brain endocrine axis, gut heart axis, gut lung axis, gut liver axis, gut pancreas axis, gut bone axis, gut muscle axis. So you can see what are the diseases that can happen in case of dysbiosis of gut microbiota. So the question was talking about if gut health is linked with gut microbiome, which is very true. It is linked with the gut microbiome. So we'll stop here today. It's almost 2 hours. So anybody has any questions from the portion that we covered today, or else we can conclude the session, and I'll take the next session in the next minute. Okay. So nobody has any questions. So I'll stop presenting, and we'll meet again next Friday, same link.